Hello, everyone. I would like to welcome you all to this month's Biotech Talk that is being hosted by the UWBC Gene Expression Center and 10x Genomics. The Biotech Talks, this is this month's first Biotech Talk. They'll carry through December. The Biotech Talks happen on the third Thursday of the month, starting at 1:30 p.m. in the afternoon. In November, there will be two Biotech Talks that will be given. So check those Biotech Talks out on the UWBC website. My name is Sandra Splinter-Bondurant, and I'm the director of the Gene Expression Center here in the Biotechnology Center. In the Biotechnology Center, the GEC is one of the 7 resource cores that are available to campus investigators to assist them with their research. The Gene Expression Center offers a wide range of RNA-based application services, ranging from RNA extractions, RNA QC, bulk RNA library preps for both standard and long read sequencing, as well as single cell or single nuclei library preps, in addition to spatial transcriptomics. Today, I'm excited to announce the new 10x Visium CytAssist spatial transcriptomic services that we will be offering to enable investigators to survey larger tissue sections that will have more probes targeted for a mouse or human tissue section. In the past, we've collaborated with the TRIP lab on the tissue sectioning and slide prep for the spatial transcriptomic assays. We will continue to collaborate with the TRIP lab on this service, as we feel it's important to let the experts do what they do best, and that would be the TRIP lab's histology section. I'd also like to give a plug before we start today's talk for the Badger Connect Research Services Fair that is being held in the Biotechnology Center on October 26th, starting at 1:30 in the afternoon. The fair will be kicked off by a talk by Dr. Hernandez, who is an assistant professor in medical physics. This will be followed by various panel discussions and workshops, and then closed with a networking reception. This is being hosted by the UWCCC, the UW School of Public Health and Medicine, as well as the Biotechnology Center. For more information, you can check out the flyers around campus on these posters, or you can go to the respective websites to find out more information. So today, with further ado, I'd like to introduce Leilani Marty-Santos, who is a spatial science and technology advisor at 10x Genomics. She's here today to give a talk relating to the Visium CytAssist platform, where she'll be sharing how bridging histology and genomics is possible. So thank you.
Thank you for that introduction, Sandra. So as Sandra mentioned, I'm your spatial science and technology advisor. And so what this means is that I'm here to help talk to you about your experiments, the feasibility of them, and what you can do with our different spatial assays. I think a lot of people know 10x as a single cell company. And so I think most people know of us from our entire toolkit, where we talk about our 3 different platforms: our Chromium single cell, Visium Spatial, and our newly launched Xenium in situ platform. Most people really know us from that Chromium single cell side. When I talk about Chromium and the fleet of single cell solutions, I know that a lot of people have really worked with this to be able to either focus on individual cell types and states, being able to study things as broad as 1 million cells, or being able to focus on something as 100 cells, so maybe look at a specific antigen. People use these assays to look at CRISPR, multiomics, and other approaches. And once again, I think most people know us as that single cell biology company. But with that, and as part of our whole transcriptome assays, a few years ago, we launched our Visium platform. And so Visium really allows researchers to combine that spatially resolved whole transcriptome data with the ability to look at a high-resolution image. When I talk about Visium, I actually really like to show this particular animation. And so what you're seeing here is the ability to take those plots that you may be used to seeing in that TSNE or UMAP format, understanding that single cell information, understanding the clustering of those cells, and being able to understand how they break apart. But being able to also put them into that spatial context is going to be incredibly valuable for a lot of researchers. I think I'm a little bit biased to this particular animation because I'm a developmental biologist at heart. I actually spent a couple of years of my postdoc here in the cell and regenerative biology department. But whenever I look at this, I always think that no cell is an island. We understand how a cell develops, differentiates, and continues to work in the body based on how it interacts with other cell compartments. So I think it's really important to have that additional understanding. I'm very briefly going to talk about what we call our direct placement Visium platform. And so when we talk about direct placement Visium, what I'm showing here is the ability to, as the name implies, directly place your tissue sections onto these 4 different capture areas. Those 4 different capture areas are 6.5 by 6.5 millimeter capture areas. This is a technology that has been in place already here at the core for a while. But what it ultimately breaks down to is being able to have a poly-A-based capture methodology. So if you're working with non-human, non-mouse, this would be the way to go, using fresh frozen samples that ultimately relate back to a specific portion of that capture area by using the spatial barcode. So consider this your X and Y coordinate on that slide. So you're capturing each transcript by using this poly-A methodology. You have a unique molecular identifier identified here as a UMI. So that lets you know if there's any duplication events or things that may happen when you're doing your library preparation. This is how you're able to tell where a specific transcript is on that capture area. Current resolution is 55 microns. Our 6.5 by 6.5 millimeter captures have about 5,000 barcoded spots. And when you begin to look at that initial workflow that we had, so this is our direct placement, not the site assist workflow. What you had to do was to be able to section those tissues onto that 6.5 by 6.5 millimeter capture area, which is where, for example, Carla's core came in because that section could be very challenging. From there, you would do your H&E staining. You can do imaging. So all of this is done on that same capture area, which allows people to be able to really identify that visual of that same tissue that they're then doing sequencing on. From there, a little bit of molecular biology at the bench to do the barcoding and library construction. From here, you would then do your sequencing. And once you get your FASTQ files back, you run it through our pipeline that does an alignment. So what that does is it takes your image, it takes those spatial barcodes, and it tells you where each of those transcripts is. From there, you can do your analysis either using third-party community developed tools. I think the most common one that I've seen users working with is still going to be Seurat, but you can also use our Loop browser software. Now, this is a free to download software, and we have a lot of different data sets, whether it's from our direct placement assay or whether it's from our site assist assay. And so what you can see here on the left-hand side is the ability to do your visualization as this video starts over again, is going to be the ability to really zoom in on a particular region of tissue, do your whole transcriptome analysis, so being able to look at that differential expression. So let's say these red spots are going to be different from the blue spots adjacent based on the genes that are being expressed, but also have that whole tissue information. So you don't have to choose a region of interest a priori. However, if there is a region of interest that you want to focus on, you have the ability to go in, select that area. Potentially, you could use this to map and mine data for different projects in your laboratory because you're able to choose those different areas, but you always have that whole tissue section to go back to in terms of information. Now, we've had a broad variety of applications that our people use Visium for. I'm showing a small smattering of them here. I think the most common ones are probably going to be that oncology, immunology, neuroscience. I will mention we've had researchers working with plants and other different species as well. So if that's of interest, please feel free to reach out and I'm happy to walk you through how to do them. But I did want to give a couple of examples of what people are doing with our technology. Now, this is actually an example from our R&D team when they were developing the technology. We have a lot of neuroscientists in our R&D team, and so in this case, what they were looking at was using single cell and bulk analysis initially to look at this amyloid precursor protein overexpressing mouse. Now, this is a mouse model for Alzheimer's disease, and so what we can see here is when they're looking at that single cell and bulk data, there's one specific gene that comes out when they're looking at this, comparing their wild type versus these transgenic replicates. That specific gene that they're looking at is promelanin concentrating hormone. Now, if you know about this particular hormone, you know that it's involved in regulating sleep-wake cycle. And normally when we look at it now in that spatial context of being able to understand what is happening and what it relates to, is when we look at that wild type mouse brain, we can see that's really only located in the hypothalamus. And so that's where the normal expression would be, which makes sense. Hypothalamus is also associated with regulating that sleep-wake cycle. However, when we look at it in that transgenic mouse that's overexpressing this protein that's also overexpressed in human patients, what we can see here is that not only do you have this very high level of expression compared to that wild type within the hypothalamus, but you can see how it's also impacting the cortex and other areas of the brain. And so being able to understand that also helps you understand that when you're looking at an Alzheimer's patient, how you can sometimes get those neurocognitive defects, not only associated with defects in their regulation of the timing of their sleep-wake cycle, but also in a lot of the cognitive function. Now, because Visium is a whole transcriptome assay, one of the really cool things is that you can use that whole transcriptome as your controls as you're working with this. And so we can see something like neurogranin, which is going to be this pan-neuronal marker, usually focused more in the hippocampus and in the cortex. And when we look at that, we can see that there are no changes between the wild type and the transgenic model. So you can really go in and begin to look at those different transcriptome-associated pathways associated with your disease of interest and see what remains constant, what changes, and the spatial localization, which we know is going to be very important for things like the brain. This is another example of work being done. So this is from Holger Haynes' group. And what they actually did, this is from Barcelona. And in this case, they're trying to build this single cell tumor immune microenvironment atlas. They started with single cell, and they actually did this massive experiment where they were working with 217 different patients across 13 different cancer types, ultimately working with some samples of about 500,000 cells. And what they were trying to do is really understand how that microenvironment looks like. So when you have different immune populations that can regulate cancers differently, how can we use this to understand what is happening in one patient versus the other, and being able to understand how that ultimately changes prognosis? And so they did that massive assay. They began to really look at the immune cells within. And so you can see how this is differentiated based on different cancer types. And you can see those different immune cell populations based on those different cancer types overall. Now, they then took the single cell information and they overlaid it on a Visium sample. And so what you're seeing here is a breast cancer sample. And so ultimately, they're looking at where these immune cells are related to that breast cancer sample. Now, what you're not seeing is the underlying H&E. But on top of that H&E, what you can begin to see are these fibrotic regions. That makes sense if you think about a breast cancer sample that normally has a lot of fibrotic encapsulation. So you'll have a lot of these extracellular matrix markers like collagens, laminins, and things like that. And then the other thing that characterizes breast cancer is going to be its hormone status. So do you have estrogen positive regions? Do you have HER2 positive regions? And so on and so forth. And so by using that genetic information that they have from both the single cell data and Visium, we can now see that this is going to be an HER2 double negative region. We have here this HER2 positive region, and then we have this HER2 estrogen receptor positive region down here. Now they took this one step further and then overlaid the immune cell information. And so this is where you're really able to understand the context of where those immune cells are matters. And so if I begin to look at this assay, we can begin to see, for example, these particular CD8 T cells before they're pre-activated. You can see particular macrophage populations. And one of the things that I really like to point out is you can see the correlation with where that hormone status is as well. So you can see, for example, having this double positive region down here in blue. We can begin to look over here. We can see these CD4 memory T cells, and then we can see those pre-exhausted CD8 T cells in pink here. When I go back to that HER2 positive region shown here in green, we can see that it seems like those pre-exhausted CD8 T cells really like to be in the same place as where the HER2 is as well. I'm not a cancer biologist, but being able to once again understand where things are in the spatial context of not only one cell type, but multiple cell types. So those hormone producing cells plus those T cells, maybe there's a recruitment factor there that could be further studied now that we know that there's this interaction here. Now, at 10x, what we found is that being able to integrate your single cell and spatial data sets is going to be really valuable. And so we have a lot of methodologies on how to do this. And we're actually meeting with one of the professors that's developing a lot of these methods earlier today. So I will mention that we have a lot of resources on our website. So this data integration analysis guides portion tells you how to be able to do this integration. If it's something that you want to just get started with, we also have a reference-free deconvolution tool within our Loop browser now as well. So something you can download for free, begin to work with, and then potentially move over to one of these third-party community developed tools if you want to do a full integration of a data set, similar to what I just showed from Holger Haynes' group. Now, I want to move over to kind of that main point of our discussion today. And this is going to be talking about that site assist workflow. The name of my talk is Bridging the Worlds of Histology and Genomics. And the reason we wanted to name it this is because when we talk to our Visium users, we kind of have two groups of customers. We have the people that are coming from that histology side, the researchers that are very comfortable with sectioning, staining tissues, putting them on slides and imaging, but maybe they don't have that molecular biology background that a lot of people in the single cell community have. A lot of our other researchers come from that molecular biology side where single cell to them is something that they do every day. They're used to doing library preps, but maybe they've never really worked on tissue before. And so what we decided to do was create the capability of an instrument that would integrate both of those workflows. Now, the idea behind CytAssist is that it's an enablement tool. It's meant to simplify workflows and give you an expanded ability to work with your samples. Now, for those of you that were present at the very beginning, I was talking about that direct placement workflow. You can imagine sometimes being able to have to section down to that 6.5 by 6.5 millimeter capture area. It can be very challenging having to place that section precisely in that area, things like that. And so being able to work with CytAssist now means that you can start with a plain glass slide as your input. So you don't have to go directly to the Visium capture slide. You start with a plain glass slide instead. This can be fresh frozen, thick frozen, or FFPE samples. You have the capability of doing H&E or immunofluorescence on that plain glass slide. And what that means is that if you have a particular section of interest and maybe you're working across serial sections across multiple slides, you can now do your screening using these staining methodologies ahead of time, which is very useful if, for example, you're working with a rare cell population. You can now say, out of these 5 serial sections, section number 3 is the one that has most of that particular rare cell population, and I know that I can move forward with that. Whereas with the original Visium direct placement workflow, you had to kind of guess where you were in the tissue and hope that you were putting the correct section onto that Visium direct placement slide. And so beyond that choice of best tissue section now, you also have that flexible tissue size. So when you look at this image, this blue box here is going to be that 6.5 by 6.5 millimeter capture area. We've also added an 11 millimeter by 11 millimeter capture area for people that are working with larger tissue samples. And you can even have a tissue section that's larger than that and then focus in on these area sizes. Now, this is our key to our CytAssist design. You can see that it's very similar to that initial Visium workflow, but ultimately this is going to be a slide that you're transferring analytes to. Now, when I talk about analytes, a lot of our workflows for CytAssist are going to be probe-based. So this focuses on human and mouse because we're generating these whole transcriptome probe sets. But we create those probes with a synthetic poly A tail. And that synthetic poly A tail is going to be captured by that poly DT that I showed earlier. So it's still going to be the same idea overall, but you're using the CytAssist to do the transfer of the analytes from the plain glass slide. Workflow is also very similar to what I had described previously. So you would start by prepping your sample on that plain glass slide. And while I'm showing an FFPE sample here in terms of the wording, this still applies to whether you're working with fresh frozen, fixed frozen samples, or other sample types. If you have questions about those, please feel free to reach out to myself or Marie. We're happy to help with any questions that you have. Now, after you do your sample preparation, which in the case of FFPE, that would be your standard deparaffinization series, then your imaging. So you could do H&E staining or immunofluorescence, like I mentioned, take a picture of that slide, and then do a probe hybridization. After you do that probe hybridization, you'll use the CytAssist instrument to transfer the analytes these probes from that plain glass slide onto the Visium slide that I showed earlier. And then from there, it's very similar to what I discussed previously. Now, instead of going to a cDNA process to be able to generate your library, you're going to do a probe extension instead. But ultimately, you'll get your FASTQ files back and do that same arrangement on the pipeline where you overlay your sequencing data onto the image that you're generating. Now, a better view of what is happening on that CytAssist instrument is probably what I'm going to show here. So you start with 2 plain glass slides with your tissues on them. These are slides that have been sectioned, stained, you've already imaged them, and then you've done that probe hybridization that I've mentioned. You also have your Visium slide with 2 capture areas, and then you place these on instrument. So what you're seeing at the top are those 2 plain glass slides that have those probes hybridized to them. These have been restained with eosin to allow for easy placement within the instrument itself. You see the Visium slide here at the bottom where you then have these two reagent holes. So that's where you're placing permeabilization reagents to allow for movement of the probes from your plain glass slides onto the Visium slide. Now, I very unscientifically explain this as being similar to a panini press, but I think that helps with the visualization. And so what you can imagine is as you close the instrument, you're going to bring those slides into close proximity. And so that's where you're going to allow for the transfer of the analytes from your plain glass slide onto the Visium slide. And you can see a cartoon of this here where we can see how these probes are moving from that FFPE tissue on the plain glass slide and are being captured by those capture oligos that are present on the Visium slide. Now from here, you'll continue to work with that Visium slide, and this is really that first point where you're using 10x reagents from the probe hybridization onwards. So let's say if we're doing the previous part of the workflow and we realize at that imaging step that maybe this slide is not the one to work with, you're basically losing maybe a super fast slide, which costs about 10 cents, and your tissue. But that means that you're able to really focus this experiment on a tissue that means something biologically to you. And then from there, one of the other nice things is that you're able to use this process to generate an additional set of images. So you'll have your nice high resolution image, but then you'll also have an image taken by the site assist from below, where you can now see where the tissue was specifically and have this spatial frame added to it that tells you the orientation of that tissue as well. Now, just to give a comparison of those different sizes, these are needle punch biopsies that were done on a human breast cancer sample. And while you can see a lot of cluster information here, you can really see different areas of the tissue. When I show you the larger tissue size, you can really see how sometimes you can miss information depending on what tissue sample you're working with. So maybe you're working with a mouse brain, the 6.5 by 6.5 might be enough for you. But if you're working with a larger tissue section and you want a little bit more information, I think this really shows the value of being able to see additional clusters and additional information in a large tissue to be able to understand what's happening. One of the really nice things about the site assist is that, as I mentioned earlier, we're really trying to enable our users to move forward with these workflows. And part of that is the ability to even just align your target tissue and choose the regions of interest that you want to work with. So in this case, I'm showing a 17-day mouse embryo, so pretty large tissue sample overall. And so what you can see here is that we have these guides. These are shown in color here just because of presentation purposes. On the instrument, you would have these thick gray bars and then these black lines at the corners. The blue bars, which will be gray on your instrument, are going to be the ones that help you align to your 6.5 by 6.5 millimeter capture area. So you can see here how we're choosing specifically either the mouse head or the mouse embryo torso in one case or the other. So these are serial sections. Or you can use those black hatch marks at the corner to allow you to align to that 11 by 11. So you can really be able to move that slide around and choose the area that you're interested in. If you have something even larger than this is where I would talk to the core, for example. You can outline the back of the slide using a green Sharpie. And I say green because normally there's a green light that's shining out here. And so that won't interfere with your image capture, but that will help the core, for example, be able to align to your area of interest as you go through. And here's the data that was generated looking at those specific regions. So what you can see here is the ability to look at that mouse embryo head. And on the left-hand side, you have that total transcriptome whole differential gene expression once again. So you're able to pull apart different areas. But then you can also look specifically for particular genes of interest and really see the differential expression of those genes alone. So we can see something for labeling the eye, the brain, the liver, or extremities. And you can really see how those genes are differentially expressed across the tissue as well. Once again, being able to get that at that whole transcriptome level. Now from here, I want to talk about some of the things that we've been working on and developing within 10x. So things like expanding sample access. I mentioned that not only do you have the ability to work with larger tissue sections, but you also have the ability to work with different tissue preparations. We're adding the capability to work with additional analytes, and then also being able to work with higher resolution in the near future as well. So for our platform, one of the things that we wanted to do was not only have either FFPE or fresh frozen as a capability. If you're working with human or mouse, you can work with any of these tissue types on site assist because this is a probe-based approach that has the whole transcriptome information. So if you're working with things like blocks, tissue sections, this is something that now allows people, for example, to have a collaborator ship them a tissue section, because sometimes you can send slides, but depending on what your IRB is, maybe you're not able to ship that precious patient block. And so this really allows people to be able to do a little bit more work. The other thing that you can do now is archival slides. So we've been testing slides in-house that have already been section stained, hard mounted, and cover slipped. And so things that have been sitting in your lab that you really didn't think were a possibility to work with now could be. I mentioned earlier that we also have more availability to work with frozen samples. A lot of assays focus on fresh, unfixed samples because that gives you the highest RNA quality. It's an artifact that just tends to happen when you do fixation, whether it's PFA or whether it's formalin, that partially degrades your RNA, usually more towards that poly A tail. So for those of you that may be working with neuroscience samples, a lot of those brain samples tend to be perfused or have a lot of different preparations. So if you have done any fixation, you can now work with fixed frozen samples as well. And you can also start with that section on a slide. So if you have a collaborator that is excellent at sectioning, they can always give you a section and then you can move forward from there. One of the things that we found with CytAssist is that we really are able to see a lot more than even when we're working with our direct placement technology. So in this case, I'm showing some comparisons with human breast cancer samples. So these were placed from the same block, but one section was placed on our direct placement technology. The other was done using the CytAssist technology. And so what you can see is that you have enhanced clustering. You can see some regions in the CytAssist assay that we're missing from that direct placement capability. And you can also see how you have even more sensitivity when looking at an individual gene in this case. And this is due to the fact that instead of having to permeabilize through a tissue and capturing on the capture oligos underneath it, you're now just doing that simple transfer of those analytes over. And so that really allows for that increased sensitivity and specificity. This also opens up access to samples that have lower RNA quality. So for our FFPE-based assays, we usually talk about a DV200 of 50% if you were working with direct placement. We found such higher sensitivity and specificity that we can go down to 30% of DV200. And what we're actually moving towards is that FFPE samples will, in the future, only be done on the CytAssist. Now with fresh frozen, what you can see here is you really are able to get good results with the CytAssist, even when you're working with a sample of a RIN score of 4.4 compared to our original recommendations of 7. So once again, sensitivity and specificity are some of the reasons that we're moving towards the CytAssist for a lot of our approaches. You can also have the capability to work on tissue microarrays. So people that are doing a lot of those needle punch biopsies for different tissues, you can actually run different tissues on the same capture area and be able to pull apart those results. So you can see this alternating tissue microarray where we're able to work with brain and kidney samples. And so you can see these in alternating order here. Not only are you able to pull apart the brain and the kidney as being different samples, but you can also go in and actually see the individual spatiality within those clusters. And I always like to focus on this specific one because it's a brain cluster and you can really begin to see where those hippocampal neurons are based on different populations, which to me is very striking. Usually when you're working with the needle punch biopsy, it's a little bit hard to be able to pull apart those different areas. So being able to understand where those tissue compartments are, where different cell types are, is going to be something that's very valuable to researchers, even if they're working with these small needle punch biopsies. In terms of other current capabilities, I mentioned that we're expanding analytes. And so what I'm showing here is the ability to get multiple data types from a single FFPE section. So I had already talked about being able to stain that same tissue section with either H&E and then go into that RNA expression, which is what I'm showing here in the first two sections. Now you also have the capability of doing protein. So we've introduced a barcoded antibody assay to the CytAssist. This basically just adds one additional overnight hybridization. So you have that hybridization with your probes. You'll then do another overnight where you're allowing your antibodies to bind to your tissue. And then you're able to get multi-omics from one tissue. And I will mention that I'm showing an H&E example here, but you can also do immunofluorescence to add additional antibodies as well. Now, this is being done because we actually created our CytAssist slides to have an additional barcode capability. And so because we have this particular capture sequence here, you can have a barcoded antibody that can then be recognized by that capture sequence. And so the way that we've launched this initially is working with our particular panel here. So this is an immuno-oncology based panel. You have 31 antibodies that are specifically focused on either tissue compartments, so things like stromal cell markers, epithelial cell markers, and things like that. But we also have a broad variety of immune markers because a lot of our researchers have wanted us to give them that capability. And then you'll have 4 IgG controls as well. But from there, you also have the capability of spiking in antibodies. Now, if you're working with 1 or 2 antibodies, I maybe would suggest being able to do something like an antibody staining, so immunofluorescence, prior to doing your cytosys workflow. So that way you don't have to do all the titrations and things to be able to incorporate them in this panel. But we are also working with BioLegend to be able to generate more barcoded antibodies. So let's say if you want to add 5 antibodies to this panel, you could add those in pretty easily and be able to do a 40-plex run basically of antibodies plus your whole transcriptome data plus your imaging data, all from one tissue section. The last thing that I want to talk about in terms of current capabilities or things that people are currently moving towards is the ability to use our probe-based assays to then add in and customize their workflows as well. So we have a tech note that really describes the ability to work with this, both for our Chromium single cell gene expression flex. So if you've worked with any fixed samples, you would use that probe-based approach, and you can design probes for single cell and for Visium to be able to cover additional information that you're interested in. A lot of people are doing this to look at microbial RNAs, be able to look at viral transcripts, things like that within the tissue. We're adding capabilities to do this with our Xenium platform, which I'll touch very briefly at the end of my talk. But one of the reasons people are doing this is because they want to be able to analyze multiple things on a tissue section. So this is from SciLife. And so what they did here was they took that whole human transcriptome probe set that we normally work with. And what they did was they actually spiked in probes against 10 genes of the SARS-CoV-2 virus. And they used this to look at the lung sample in patients to be able to understand what is happening and how things are changing in response to viral interactions. And so they did the comparison where they did, human only, SARS-CoV-2 only, and then they did the colocalization. I'll show some of the colocalization data here. But one of the really interesting things was when comparing human only to the colocalization, they were able to find some very nice differentially expressed signals associated with infection that were dominated by myeloid cells, endothelial cells, B cells, epithelial cells, and fibroblasts, which to me pretty much encompasses the whole lung population, but they're really seeing a lot of changes across the board. Really interesting to be able to understand what is happening with those myeloid cells and plasma cells within the tissue. When we actually look at the samples themselves, we can begin to see things like Col3A1. Those are going to be markers of the fibroblast population within the cells. We can also see AT2 cells that are also being affected. And so we can see changes in these populations when we look at the control versus a COVID-infected samples. Fibroblasts are particularly interesting to me just because we begin to lose that capability to have, that breathing capacity, because you're going to have a stiffening of the lung. And that's something that we've seen with COVID patients, and also just ability to create surfactants and things like that, which are in the domain of that AT2 type cell. So we can really understand what is happening within these cells and being able to understand what could happen potentially looking at this at different intervals. So maybe you have a patient that had a short COVID response versus a long COVID response, and now really be able to see not only what is happening, so those differential gene signature expressions that we could see with single cell, but also understand where in the tissue they're happening. So how close are they to a COVID source of infection, and then move out from there, for example. Now, the last thing that I want to talk about with CytAssist is an upcoming capability. So this is what we call our Visium HD. So Visium HD, instead of being that 55 micron resolution that I talked about throughout most of my talk today, which is where we're at, we're calling this our standard definition Visium. With HD, you're going to be able to get to that single cell scale. So we're going to be working with 8 micron resolution total. This is actually going to be a capture grid of 2 microns by 2 microns that you're then going to bin up to 8 microns for analysis. And I have an image of this right afterwards. Now, this is only going to be on the CytAssist. And this is due to the fact that we have that higher sensitivity and specificity. There's less risk of transfer or diffusion when you're working with the CytAssist, and that's something that we've tested in-house very robustly. You have that capability, once again, of starting with a plain glass slide, doing H&E staining, and then being able to get to this high resolution view. It is going to be only FFPE at launch, but we are working on additional capabilities. And then it's going to be 6.5 by 6.5 because you're just going to have a lot more sequencing associated when you have these much smaller areas. However, this is what it's going to look like. So instead of the space between those different spots where a lot of people have been upset, they don't want that white space there. We've heard our customers and we've re-engineered our slide. And so what you can now see is this nice grid structure instead. And this ultimately breaks down into these 2-micron squares. So basically barcodes are going to be changing every 2 microns. But when you're sequencing, that can lead to a little bit of sparse data. So what we're recommending is going to be this 8-micron square area for binning, but that's just going to be your features. And we're also going to allow the capability for those people that have that bioinformatics knowledge and want to bin in different forms, they can do that as well. This will ultimately be able to get you to very high resolution data. And some of the examples that I've seen from our R&D team are really astounding. And so what you can see here is the ability to look at that H&E image, then look at that whole transcriptome information to get your unbiased clustering, and then ultimately break down and be able to see individual cell types. And so I can show here LYZ1, which is going to be this PANF cell marker that's at the base of the crypts. This is a uniform cell type. To me, it's not as exciting as when I show these MUC2 labeled goblet cells. You can really see them interspersed and you can see where they start and stop basically with this grid. Now, the other image that I love is looking at this breast cancer tissue sample where we're comparing fibroblasts with other smooth muscle cell producing cells. And so you can really see how you have this nice level of information. You really have this broad clustering pattern once again across the entire tissue, but you can really focus, and this almost looks like immunofluorescence to me instead, but this is actually going to be based on those barcoded spots, or I guess grid squares now. And so you can really see that nice overlay and understand once again where one population of cells starts, one population of cells stops, and how they overlay overall as well. So hopefully we've taken thus far from my talk just how much CytAssist really enables spatial transcriptomics. So we've improved that gene sensitivity and specificity. I talked about this and showed examples of fresh frozen, but we also have data showing this on FFPE samples. We've expanded our tissue type compatibility. So what I mean by this is because you're starting with that plain glass slide, you can now work with samples that you have in the laboratory, potentially working with archival samples, or maybe just a section that a collaborator down the hall can give you instead of giving you a block. You can also work with fresh frozen, fixed frozen, which is something that a lot of other technologies don't have the capability for. You have improved spatiality, and so I showed that example where you can compare the fresh frozen sample from direct placement to our CytAssist workflow, and you can really see those nice clusters overall. It's an easier workflow, so you could have someone that's really dedicated to your histology doing the beginning part of the workflow, have a great handoff point and be able to do the genetics part of the workflow and then go from there. We have the capability for multi-omics because we have that spatial proteogenomics tool being able to look at antibodies on that same tissue at the same time. So getting highly multiplex data from whole transcriptome and from protein and your imaging data on the same tissue. And shortly we'll have Visium HD. So that's something that I'm really excited about. And so that should be launching at the beginning of 2024. Now, for the very last portion of my talk, I want to talk about our Xenium platform. So this is going to be our recently launched tool that allows for single cell or actually subcellular resolution when looking at imaging data. Now, by contrast to both Chromium single cell and Visium, this is not a whole transcriptome assay. This is going to be a focus approach. So you're working with panels currently of about 300 to 500 genes. We will be launching a 5,000 plex capability next year. And instead of being a sequencing-based platform, this is an imaging-based platform. But it really allows you to get to that subcellular resolution. It's high throughput because you're working with a larger tissue capture area and is compatible with a lot of different sample types. Now, to show the power of Xenium, I'm going to show this very large human breast cancer tissue section. And what you're seeing is part of the non-destructive methodology of this in situ platform. And so after Xenium, you can do H&E staining, you can do immunofluorescence, or I'll show you in a little bit that you can actually do CytAssist as well. So you can get to both subcellular resolution and your whole transcriptome data, albeit at a bit of a lower resolution on the same tissue section. But then from there, what I'm going to do is I'm going to overlay clustering data that was generated using that about 300 plex panel. In this case, this is a 313 plex panel. So 313 different genes. And you can see how you're still able to generate differential expression patterns. So you can see that, for example, these blue and teal cells over here are going to be different than the pink cells that are adjacent to it. But let's zoom in and actually see what's happening within the tissue. So now you can see how we've taken this not only to that single cell resolution, but we've actually taken this to subcellular resolution. And what you're seeing in this very last inset here is going to be different transcripts that are associated with different cells. Now, because even 313 transcripts at a 50 nanometer resolution is a lot, what I'm going to do is I'm going to take away some of those transcripts and we're going to focus on a couple here. The ones that I'm focusing on are CD45, CD3, and CD8A. And these are of interest because they're labeling a rare cytotoxic T cell population that's infiltrating the tumor cells that are shown here in pink. So these green cells are the cytotoxic T cells, and then you have these tumor cells in pink. And you can really see where those transcripts are located. This technology will give you an XYZ coordinate for each of those transcripts, once again, getting you down to 50 nanometers instead of 55 microns, which is where our current Visium is. And so for people that are interested in this approach, it's something that might be of interest. It's meant to be a very easy workflow. So for anyone that is familiar with histology, you're going to be doing your normal tissue processing, so sectioning onto our Xenium slides, doing traditional tissue preparation, followed by a probe hybridization step. So you'll be doing multiple probes per transcript target to allow you to be able to visualize a broad dynamic range. So things that are high expressing, things like beta-actin, GAPDH, insulin, or things like that. You can also visualize things that are very low expressing because we'll tune the number of probes per target. I'm showing the example of one single target here. And then from there, from the circular probe, you'll do a ligation that fully creates a circle. Now, this ligation is going to be highly specific. And when I talk about highly specific, we're down to the level of a single nucleotide. So we are already working with users that are designing probes to different isoforms or different gene fusion events. But what we've shown in AGBT and in other events is that you also have the capability of getting down to single nucleotide variants. And in the future, we're also working with TCR, BCR detection as well at this 50 nanometer resolution. Then from there, you generate this rolling circle amplification product. And this is where you go onto the instrument, and this is going to wash on different cycles of fluorophores that are going to bind to those probes. And that's going to allow for your transcript detection, which is what I showed in my previous image. Now, as I mentioned, Xenium is the only non-destructive methodology for in situ platforms. And so what you have the capability of, as I mentioned earlier, is doing post-Xenium H&E. So I showed that with that breast cancer sample, and this is of use for people that are collaborating with a pathologist, for example. You can send them that H&E image. They can do an unbiased annotation, and then you can correlate that with different data sections and be able to understand where genes are expressed. It might correlate to different changes in the morphology of the tissue that the pathologist can view. You can also do post-Xenium IF staining. So you want to be able to have that multi-omics capability. And we will be adding barcoded protein to this platform as well in the future. But currently you can at least do immunofluorescence to be able to visualize your tissue. And then, as I mentioned earlier, you can use that cytosys that I've been talking about to be able to get to whole transcriptome information from a tissue section. An example of this came from a recent preprint that we released late last year. And so in that case, we were working with a particular breast cancer sample that had initially been identified by a pathologist as being double positive. So we talked about breast cancer being related to hormonal status. So in that case, it was HER2 and estrogen receptor positive that it had been identified as. When we ran it with our standard breast panel, so that 313 plex panel that I showed earlier, we actually were able to identify that it was triple positive. So being HER2, estrogen receptor positive, and progesterone receptor positive. This was only in particular regions of the tissue though. So what we did was we actually transferred those regions using cytosys, and we're actually able to identify 94 differentially expressed genes in those triple positive regions. So if you've ever had this happen, where I think it's pretty common where you get an unexpected result when you're working with your samples, you have that capability of going back and interrogating at that whole transcriptome level as well. Now, the last thing that I want to show is the ability to work with our software. So just like Loop Browser, this is something that you can download for free today and be able to work with from your laptop. I regularly run this from my own laptop or any other computer. You don't need a higher powered computer. So software is free. And we also have about 17 sample data sets across different tissue types that you can work with. And so ultimately what will happen, there we go, is that you'll be able to use this as similar and as easily as Google Maps to be able to zoom in, pan in on the tissue. You can see how you can get to very high resolution. Now you can see how we've generated cellular outlines, and just by the click of a button are also able to visualize where the nuclei are. And so what you can do is you can imagine being able to overlay transcript information like what I showed earlier and be able to get that information. We've also allowed for different views. So on the left-hand side, I'm showing these density plots to be able to look at transcripts across the entire tissue. You can also do the unbiased clustering that you're used to doing with single cell biology, but put it onto the tissue itself and understand that the pink cells that were shown were different from blue cells adjacent based on that differential gene expression. And then on the right-hand side, I'd like to talk about the fact that sometimes simple is better. Maybe you just got your data off the instrument and you have lab meeting tomorrow and you want to have something to show for it. You have the ability of doing something as simple as using our lasso tool to be able to circle different regions of interest. I can easily imagine looking at a cancerous lesion versus a normal margin and looking at the differential transcripts between them, taking it out into something as easy to use as Excel to be able to do that analysis, or maybe R, Python if you have those capabilities, but then you're able to do that visualization very easily and move forward from there. So with that, I've talked a little bit today about our different toolkits. So Chromium single cell technology, major focus on our Visium CytAssist platform, and a little bit about our Xenium platform. But I really want to be able to highlight the fact that these tools are very synergistic and allow for people to be able to get to their information. We now are really looking forward to seeing these techniques in your hands, especially that CytAssist instrument that just launched. And so I really want to be able to talk to you further about what unique perspectives you'll gain from that and what information will be useful for you. So with that, thank you for your time, and I'd be happy to take any questions.
So that's not just for human use, right? You do it for anything.
Fantastic question. I'm going to repeat it just
Yeah.
because for the Zoom audience. So the question was, with Xenium, you're designing these targeted probe-based panels. Can you do these for species that are non-human, non-mouse? And the answer is yes. So we've been working with people that are working with a broad variety of species. As long as you have a reference transcriptome to align to, we can help you, and you're not designing your own probes. We help you with that workflow. So we have our applied bioinformatics team that will be doing that design. We'll basically ask you for your list of genes. If you have an ensemble ID that's associated with it, that makes things a lot easier for us. And if you have single cell data, because that allows us to do that tunability of probes that I mentioned earlier. So, just to be able to understand that if you have beta actin, maybe we design 2 probes to that versus 10 probes to a very low expressing gene. But we've seen people work with cavefish, plants. I have a group that's doing bee brain. So we're really spanning the gamut there with Xenium. Thank you. Yep.
And for the H&A staining or the immunofluorescence that you do after Xenium, like, is there any way to do any kind of staining before anything starts, just so you can kind of narrow in on what you're looking for, like an ROI?
Fantastic question. So the question is, instead of just doing the post-Xenium immunofluorescence or post-Xenium H&E detection, is there a capability to do staining prior to placing on the Xenium instrument to be able to detect your cells? That's actually something that we do. So you saw that DAPI staining that I showed in my view of the Xenium Explorer. So this is actually, DAPI is done throughout the run, but you also do DAPI prior to placing on instrument, because that's what will allow you to be able to look at those different tissue types. So you'll be doing it mostly DAPI based. But one of our recommendations is actually doing an adjacent tissue section. So unlike our Chromium single cell assay or even our Visium assay, you don't have to do quality control with Xenium. So you don't have to do a DV200 or anything like that, but we do recommend taking an adjacent tissue section, doing H&E or immunofluorescence, just to make sure that the tissue looks good. So no major holes, no artifacts from any bad tissue preparation. If things look good, you're able to move forward.
There are a few questions, I'll just
Yeah.
Do we have any recommendation or preference between fresh frozen or FFP when it comes to Visium cytosis?
So the question is, do we have any preferences or recommendations when it comes to fresh frozen versus FFP samples for Visium cytosis? My recommendation is usually whatever is best for your particular tissue type of interest. So if you're working with fresh frozen, I know that a lot of mouse biologists, for example, do fresh frozen samples, a lot of people that work in neuroscience as well. If that's your preferred methodology, go forward with that. If you're working with, let's say, I showed some examples of lung images. A lot of the times lung will be done in FFPE, and a lot of patient samples come in FFPE as well, just because that gives longevity to the sample. So that should work as well. Recommendations would just be working to keep your tissue as safe as possible. If you're working with cytosis, that RIN score of 4 or above for fresh frozen. If you're working with FFPE, a DV200 of 30% or above. Our usual recommendations, but I think that's kind of the major ideal overall.
And those are recommendations, right? They're not the.
Yes, exactly. I usually like to mention that those are not steep cliffs. If you do have questions about a particular sample type, let us know and we can talk to you about how to best go with those preparations overall.
One other question is how common it is now for people to actually use their single cell data and overlap it with their spatial transcriptomic data?
So repeating for the Zoom audience, the question is, how common is it for people to use single cell data and overlay it with their Visium or spatial transcriptomics data? It's actually getting much and more common. I think because a lot of people may have already been doing single cell since that's been around a little bit longer. It's something that they have as a resource. There's a lot of repositories out there, but maybe you want your specific sample type. So that's going to allow you, if you don't have a Xenium present, for example, or until HD comes out, to get down to single cell resolution so you can do deconvolution. I showed a slide where you have some of these different tools for that deconvolution. So cell to location, tangram, even Seurat is used for a little bit of that deconvolution. So that gets you to single cell resolution from a Visium assay, just to be able to get to that information from both sample types. You're welcome.
There are 2 Zoom questions.
Oh, thank you. Do we know what Xenium is going to be about? What our core here at UW-Madison?
So the question is Xenium available currently at the core here at UW-Madison? It is not yet. This is a technology that we've recently launched. Talk to your core and see if it's something that they have in mind for the future. If not, reach out to us and we can always set you up with cores in the area. We know that there's at least one in Wisconsin itself, and then there's a couple in the surrounding areas as well.
Thank you. Is there an age limit on the archive slides that are able to be used for the Visium cytosis?
Fantastic question. So to repeat for the Xenium audience, is there an age limit on archival slides that are able to be used for Visium cytosis? We haven't found it. What we see overall is more than the actual age of the slide itself or even a tissue block. It's how that block and that tissue has been stored. So as long as things look good overall, that's usually a good starting point for archival slides. So things that have been section stained, cover slipped in your laboratory. We have very specific protocols that teach you how to do a gentle removal of that cover slip, gentle removal of that excess mounting media that may be on the slide. And then you'll use a pico extraction kit and a blade just to take the tiniest piece of tissue. So like I said, it's a pico extraction. So you can imagine just how small that tissue needs to be. And that's where you'll do your DV200 measurement. Sorry. From there, after you do that DV200, if it's 30% or above, you're clear to go. Once again, I've seen people that maybe this is the one patient sample that's very precious and they still want to move forward with it. Talk to me. We can talk about, kind of how to best get to that, even if your DV200 is not perfect. But I've seen people that have been going back 10 years or more with slides and blocks. With site assist for blocks and for Xenium, we've actually seen people go back about 30 years and we're still able to get some data from them. So it really is kind of case dependent, but if you have good tissue quality, things were prepared correctly, there's good odds that you'll be able to do something with it.
So the coming question, what is the thickness recommendation for the tissue for section?
What is the thickness recommendation for tissue for sectioning? So when we talk about site assist, if you're working with FFPE, usually the average is about 5 microns. Thickness can range from 3 to 10 micron for FFPE. If you're working with fresh frozen sections, those tend to be a little bit thicker. And so people will usually do from 10 to 20 microns in those cases. Usually 10 is going to be our recommendation just because it allows for better probe penetration.
And for anyone that was here looking after we announced it, we actually have literature in the back of the room for some statistics, instrumentation, and some other tech notes. And there's also notepads and things, so feel free to grab anything on your way out.
All right, if there are no further questions, thank you everyone for your time. I really appreciate it.